Hey guys, so today, a pretty big topic. This usually is 2 weeks worth of lessons. We're going to do it all in one. So we're going to talk transcription, and we're going to talk about some of the post-transcriptional modifications that can happen to messenger RNA. So let's talk about it. First, we got to start with the central dogma. So the next 2 weeks are actually going to cover kind of the key intermediates between the central dogma. So we've talked about DNA before, and we're going to talk about transcription today. And that's going to get our mRNA, and then we're going to next week take our messenger RNA, we're going to translate it into an actual protein. Okay, and so that's what we're going to focus on. So actually today we're focusing on transcription, and so that's when we're going to actually make an RNA. So I don't want to just say a messenger RNA because we're going to make all of our RNAs this way using RNA polymerases. But for the most part, usually what we're thinking of is actually a messenger RNA that we're going to then go make a protein from. But that doesn't necessarily have to be the case. That's just typically probably the best way to learn it. Something I want to emphasize, let's back up a little bit. So when we're transcribing, we're going from two of the same molecules, okay? So DNA is made of nucleic acid, right? As is RNA. So we're just transcribing. So if you're transcribing like a note or something, or let's say you have a sheet of paper and you needed to transcribe it into another sheet of paper, you're basically copying it over. It's not exactly the same. Maybe you're trying to copy some notes, and so you have like a printed sheet from like your computer, and then you want to write out what it is. Well, you're not going to change the language. You're still using, letters. And so you're transcribing. Now, if you were going to take that and change that into something different, let's say you were transcribing a description of how to bake a cake, and then you actually baked the cake, okay? So you've changed the medium that you're actually in. So instead of transcribing in English, now I'm going to take that transcription, I'm going to translate it into something physical. And so that's what we're dealing with the central dogma. But today we're talking about transcription. Transcription itself is really, really interesting. We're going to use an RNA polymerase. We're going to talk about, there are a couple different kinds of RNA polymerases. We're going to talk about that. And we're going to just kind of zoom in on what's actually happening. Now, we've talked about replication before. It's very, very similar, okay? So in replication, we open up our strands, right? And then we're going to build from the 5 to 3' direction, right? Because our carbons for our sugar phosphate backbone are still oriented the same way, and we're really not doing anything that different from replication. But the enzyme is just a little bit different. It's still a polymerase. It's still going to make a polymer of nucleic acid. In this case, we're just going to make a complementary RNA transcript as opposed to a DNA transcript to this size, or this side, sorry. One of the key things I want you to understand is sense versus antisense. This is a common question that gets asked. This is also coding and non-coding strands. Make a messenger RNA. This piece of DNA here, okay, so it's just going to keep on going. My messenger RNA is going to look exactly like that. The only difference is that instead of T's, instead of thymines, I have uracils, right? And we know that those are very, very similar molecules. The only real difference is a methyl group. Otherwise, they're almost identical. And so then, see, the A's are the same, so adenines are the same, cytosines are the same, guanines are the same, and the U's just become T's, okay? So they're almost identical. And so your messenger RNA looks just like this strand, right? The complementary strand, however, the one that has the opposite, so A's match up with U's, T's match up with A's, G's match up with C's, C's match up with G's, we call that our antisense or non-coding strand. The coding refers to the actual ability to take a piece of DNA, turn it into an RNA, and turn that into a protein. So this is the coding strand because my messenger RNA looks like that. But in order to make this, it has to be complementary to the non-coding strand, okay? Now, just to solidify things, I want to cut to a video that's just going to kind of visualize what's happening.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called Rna Polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm.
That's a really, really good video. I like that. Visually, it's awesome, and that lady has a British accent, so you know she's trustworthy. Now, something to talk about is the idea of a codon. So codons are just 3 bases in either your coding strand of DNA, so that's this guy, your sense strand or your coding strand, or your messenger RNA transcripts. So we've got our 5' end, we've got our 3' end, right? So we built this direction, we always build, so we add new nucleotides to the 3' end. And so we're going to build this direction, and we always start with our start codon. Okay? And then you can see down here, we're going to start with our amino acids. Now, if you take biochemistry, which I also teach biochemistry, then we'll talk more about amino acids and proteins, and we're going to talk about that next time too, when we actually talk about translation. But today we're just focusing on codons. And so codons are just these 3 nucleotides that code for a specific amino acid, either a start or whatever amino acids in between. We have 20-ish, though there's some variability there. And then we have our stop codon. There's a couple of those. Now I want to emphasize there are a couple different ways to actually figure out what your protein is going to be, and we use different charts. There's really 2 styles of charts. There's the style on the left, which is like the wheel-based chart. And so we know this is in RNA because it's got uracil, so it's an RNA-based chart, and you would just start in the center and then you move out. A lot of textbooks don't use this style. They like this style where you pick your first position nucleotide over here, and then your second position over here. So you see like all of these start with a C in the first one, and then all the second letters here are U's. Okay. And then on this side, you pick the third position. I find it oddly confusing. I don't know if I'm just crazy. I think this is such an easier way. So I actually have 2 styles that I like to use. Either you have your DNA, so this is your coding strand, DNA, and then you also have your messenger RNA. So it just doesn't matter really. Yeah, they're still going to code for the same stuff. It's just, do you have a T or a U? Okay, and so those are really easy questions to ask. It's really not that hard. You just take your 3 codons, and then you look at your chart. So you say AUG, and you go to your chart, and you say AUG. Methionine, there we go, and look at that, it matches up perfect. Thank you, Khan Academy. So that's how you use those charts. Pretty straightforward. Start getting into a little more of the complexity of what's actually going on. So last week we talked about genotypes and phenotypes. We talked Mendelian and non-Mendelian genetics. And just to re-emphasize, phenotype is the physical expression. And we actually can see that, and back in the day before we could understand the actual order of DNA, then we would just see the phenotypes and we can kind of go from there. But now we know that there's genotypes. There's actually the physical genes that make up the expression, right? And so we need to get our messenger RNA transcripts out into the cell so that it can transcribe those proteins and then change the physical expression of things. And so the typical structure of a single gene looks about like this, and there's some terms that you need to know before we get too deep into it. So your actual RNA sequence is going to come from here. So this is our DNA. So we got 2 strands of DNA here. Okay, and so that's what we're looking at here is it's getting pulled apart. Okay, so there's our 2 strands here. Okay, 5' to 3', 5' to 3'. Awesome. We're going to pull this apart. We've got our coding strand on top. We've got our template strand or our non-coding strand on the bottom. And we have to start transcription and we have to end it, okay? And so if we're gonna start it, we have to tell our RNA polymerase where to start, and that has to do with the promoter region, among other things. So there are lots of different pieces and effectors that are going to affect how often we transcribe and whether or not we can transcribe. And it just, there's a lot that we're going to talk about. So get ready. It's going to be fun though, because it actually helps explain how we get, at least in eukaryotes, how we get different expression patterns of different genes, because your cells are constantly picking and choosing what proteins they need to express. And so it's really, really important that we understand how they're picking and choosing, because they don't have brains, right? So it's all dependent on these upstream regions as well as downstream regions, okay? So upstream is anything upstream of the RNA, okay? Because we're gonna build from 5' to 3', right? So we're going this direction. So we're gonna start transcription here, we're gonna head this way, and we're going to terminate transcription down here. And so anything to the left, I guess, here is upstream, okay? Because we're gonna flow this way. And our promoter region is our really important region before this, okay? So that's gonna be our main region that we're gonna focus on. That's where your RNA polymerase is going to come over here, bind to the promoter region, and that tells it where to actually begin transcription. And then where to terminate is also important, but that usually has more to do with a specific sequence here in the template strand, okay? So that's just a typical structure of a gene, okay? We're talking about the actual structure of our gene and what it's going to do. We're going to make a protein from it, but first we got to make our messenger RNA, and there's different polymerases that do that. So there are generally 3 RNA polymerase types that we talk about, okay? Honestly, most of the time you're only going to be talking about RNA polymerase 2, okay? Now you can see it's not just one enzyme, it's a massive complex of enzymes. Obviously, I mean, just look at this one, there's so much going on. Now, some of these are including some of the other transcription factors that you typically encounter, and we're going to talk about all of those things, but I did want to emphasize that there are other polymerases, other RNA polymerases that do exist. Now, usually their job is generally to make ribosomal RNA. So ribosomes are not proteins alone. How do I say that? They're not just proteins. It's an important concept to get into your brain that not everything that does stuff is just a protein. Proteins can have sugars attached, they can have lipids attached, they can have other structures built into them. So ribosomes being the best example of a protein complex with RNA complexed, and it makes one giant enzyme or a ribozyme. And so we need to make the RNA that is actually part of the structure of ribosomes, and that happens in the nucleolus, which the nucleus, that's one of the subnuclear compartments is the nucleolus. There are other subnuclear compartments that have specific effects. If you take cell biology, we talk about that. But suffice to say, RNA polymerases 1 and 3, that's generally their job. RNA polymerase 3 can also make transfer RNA, so those are the funny T-shaped looking RNAs that we actually see that bridge the gap between your messenger RNA and your actual amino acids. And so it makes those things. And there's also like small nuclear RNAs that do a lot of different things. There's more than one type of RNA is an important idea to make sure is in your brain. Now, RNA Pol 2 is the one that we're generally going to be talking about. Now, they all sort of share similar structures for their core unit. And so we've got a nice protein structure here. You can see there's a lot going on. There's a lot of different domains here. But this one's nice because it's got a little diagram here. And this piece here is the piece that's going to actually clamp down. So we have like a cleft, okay? So it's open here. You see some space. It's not as dense here. And what that is the pocket. So that's the active site here that's actually going to grab onto the DNA, okay? It's going to grab onto the DNA, and it's going to grab onto the template strand. So we've got our template strand in red here, and then the coding strand is in the blue here. And so this is just a cartoon version of what's going on. It's going to slide along. So this is actually oriented differently. So this is kind of the side view, and this is from like a front-on view. So the DNA is going to fit in here, and then you can kind of turn it 90 degrees, and then it's going to slide this way. And so we're going to actually, well, the direction of the transcription, but we're building in this direction. So we always build 5 to 3. That's all I really care about. And so we're going to build our new messenger RNA, and we're tacking on to the 3' end here with 5' end nucleotides. And so the green here is our messenger RNA. It actually comes out a different spot. And so your DNA is in here, and then it kind of slides out a different spot. The actual physical structure isn't as important as just understanding that this RNA polymerase, with a bunch of other units, will actually grab onto the DNA, open it up so it has helicase activity, and it's going to open it up and actually build that nucleotide. That's the polymerase activity. Now, something I wanted to talk about is how does transcription actually initiate? A lot of genetics courses gloss over this, and so I really wanted to talk about this and talk about the recognition sites and how does an RNA polymerase actually know where to grab onto the DNA and actually make that messenger RNA. Well, it turns out there's a bunch of things involved here. And so we're just going to gloss over what's really going on, but I'm going to try and give you a little bit of detail. I know that this slide is super busy. There's a lot going on here, but this is really, really cool stuff. There's a lot happening. So the actual complex of transcription, and we're focusing on RNA Pol 2 right here, but of course the other RNA polymerases also have different structures here. But this is what we're looking at. So this is basically this. Okay. Now there are different structures that we need to talk about. So the ones that we usually talk about is your promoter. So we have our core promoter region here, and your promoter is going to have a few pieces of it in the DNA that actually look very similar. We're going to talk about that in a minute, specific motifs that we see often. But there are other pieces of the DNA that we need to talk about, and those have names. So ignoring our promoter region, which tends to have a ta-ta box, so a thymine, adenine, thymine, adenine, we're going to talk about that in a minute. There are other pieces of DNA, and so there are chunks of DNA that we give names to. And then those chunks of DNA actually have proteins that interact with them. Okay? And so you can have, and their names are really nice. So enhancers, they help, they have activators that grab onto them, and those sort of stabilize the transcription complex. And so if you have enhancers, they just kind of enhance the ability of RNA polymerase to bind to the promoter region and thus begin transcription. Okay? And so it's all about like just sort of building that nice scaffolding to enhance transcription. And so enhancers are the DNA and activators bind to enhancers. And you can see they kind of bend over this DNA here to just sort of kind of bring it all together and make it nice and happy and clustered. Now the opposite can also occur, and that's silencers. So silencers will have repressors that bind to them that can then prevent some of these bindings from occurring. And so we see repressors used more often in bacteria, but the concept of a repressor does happen in eukaryotes. And so they bind to silencer regions. Now, something important to note, these are all downstream. They don't have to be. You can also have enhancers or silencers or insulators, which I'll talk about in a second. They can be downstream or upstream. It doesn't really matter because the DNA can bend over and fold over on itself. Sometimes people get caught up in the idea that if it's not right next to the promoter, then it's not going to do its thing. But actually, a lot of enhancers and silencers can have a considerable distance away from the actual promoter region, which is really interesting. Insulators are another idea that don't get talked about that much. Basically, what an insulator does is it's a region of DNA that can sort of inhibit the enhancers. So if enhancers can enhance transcription and silencers can sort of silence transcription, then insulators are just one more level blocking the enhancers. I know, it's a lot. But it has to do with DNA methylation. And you just basically can methylate an insulator region, and then that prevents this guy from binding. And this protein, when it binds to the insulator, prevents transcription. So it's kind of like a silencer in a way. So insulators are just a subset of silencers, at least in my opinion. But hey, what you gonna do? There are lots of proteins associated with all of these things. So you have your DNA region over here, and then you would have your interacting protein. And of course, there are multiple versions of interacting proteins. Okay, proteins. So I say activator protein. Now there are a bunch of those. They can have different names. And it kind of depends on how you want to parse things out. So an activator binds to an enhancer. Awesome. Now, the other end of an activator can bind to a bunch of different things, and we call those our co-adapters or co-activators. There's different names for all of these, and there's, of course, a bunch of different ones. But just to understand the idea, the concept is more important to me, that you can have all of these proteins binding together, and those together allow the RNA polymerase to bind. They either allow it to bind or they tell it where to bind and can increase transcription. So RNA polymerase can just bind to the promoter and begin transcription. What these things can do is make that happen 1,000 times faster, 10,000 times more often. And so that's kind of what we're looking at. Now, you can completely stop it, but oftentimes what we're really talking about is, does it happen, let's just make this up, once every second? Does it happen 10 times every second? Does it happen 1,000 times every second? And your enhancers and your silencers and your insulators and all these co-adapters, those are what kind of control the rate of transcription. They don't usually completely silence it, though that can happen, but usually we're talking about enhancing. Okay? And one last thing before we get into motifs and the different specific pieces, like your ta-ta box of the DNA. Remember that all of these are proteins. And proteins are all made from messenger RNAs, which are all made via transcription. And so these proteins had to be transcribed at some point, or translated from a messenger RNA that was transcribed in the same way. And so you can see, like, if I reduce the amount of this coactivator 40, it's going to reduce the amount of this messenger RNA that gets made. And so you can see what we're really talking about are levels of control. How do I control what actually gets transcribed? Well, all of these different things can vary in the actual amount or the concentration that are inside the cell, and that's going to change how much of this thing you get to make, how much messenger RNA you're actually making. And so that's really, really interesting. So that's how, at least usually in eukaryotes is what we're talking about, how we actually control transcription, which is how we inadvertently are controlling how much protein is made, which is the phenotype, which are the physical things that we're seeing. Ah, so cool. Zoom in on this actual DNA. So ta-ta box comes up a lot. There's specific proteins that bind to the ta-ta box. Why is it called ta-ta box? Well, it's like this alternating Ta-Ta sequence, and we just call it the box because this what we've always called it. I don't know. It's the ta-ta box. It's a little box. See, there's a box. And what are these? Okay, so in the actual structure of DNA, we see consistencies. And when we were able to start sequencing DNA, we started noticing, like, it wasn't just completely random. Well, first off, you have your codons, right? So your codons code for specific things. But if you actually look at the DNA, you can see that there will be regions that have these consistencies, or they'll be palindromes. So they'll have, it'll say Ta-Ta and then AT-AT at the bottom. And you can have a really complex palindrome. So they're these repeating sequences. And so what we know now is that a lot of these proteins here will look for specific palindromic sequences or specific motifs that are consistent nucleotide orders in DNA. And there's a ton of these, and they have a lot of variability as well, but there is consistency, and that's what's really, really important. And so what we're looking at here is our promoter region. So I'm just zoomed in on this core promoter right here, okay? And so you can see we have a lot of different things. So there's your ta-ta box, so that's where a ta-ta binding protein is going to grab onto. We also have other recognition elements. You have an initiator site, so transcription actually physically starts right here. So this is, if transcription is going to start, like let's say right here, that has a specific region. Y, and so you'll see some strange, these are nucleotides, but you'll see strange things like, I think Y refers to, it's either a C or a G, or it's either an A or a T. So there's some nomenclature here that's what gets confusing. No, there are not nucleotides that are Ys. But you would have like, actually let me check. I'm sorry, I forgot. So Y is pyrimidines. So it can be a C or a T, so a cytosine or a thymine. Yeah, so that's what that means. N, any base, and then an R can be a purine, so a G or an A. Yes, is that right? Yes, that's right. I have a cheat sheet right here. See, so you can look up these cheat sheets. See, R is an A or a G, Y is a C or a T. You can have an S, which is a G or a C, so that's kind of goofy. Anyway, so that's what these actually mean. But you can see there's also downstream elements. Okay, and there are upstream elements. It gets real complicated really fast. Here's a nice looking chart of different consensus sequences that we see across different organisms. Okay, so these are different organisms over here. These are E. coli, though. These are E. coli promoters. So it's getting a little complicated because we tend to be talking about eukaryotes, but these consensus sequences can occur in other organisms, lower order organisms. But you do have your promoter region, okay, and that's what's zoomed in here. And so we see these consistencies, and look, there's that Tata box, okay. Now, everybody hears about the Tata box. It's just the most fun one to say. That's honestly why it gets talked about the most. Um, but it's just harder to say the TTGA box. Like, nobody cares about that. You could call it the cat box, I guess. Anyway, but that's upstream, generally, of your actual initiation site. And the actual transcription site initiation, so I wanted to point out, why am I showing this? Well, because I wanted to show you can, the nomenclature here. So you would say, if I'm starting at this C right here, then anything beyond that is plus. So +1, +2, +3, blah, blah, blah. And then you go backwards too. So you can have, your Tata box tends to be about 10 nucleotides backwards from the actual initiation site. That was the point of that. About eukaryotes and prokaryotes, and we need to talk about this because it's something very, very important. In bacteria, they don't have a nucleus, so therefore they don't have a nuclear membrane. Now that's not to say that they don't keep their DNA pretty organized. Bacteria can also have histones that keep their DNA. Well, they call them histone-like proteins, but they're basically histones. They serve the same function. The DNA in a bacteria is not just floating around everywhere. It's contained, it's nice, but it just doesn't have a membrane. It's not a membrane-bound organelle. And so they can take advantage of that. And so you could have your DNA strand here, okay? And you can have your RNA polymerase, which is what we're talking about. So it transcribes this messenger RNA. So in the red here, hopefully you're not colorblind, in the red here is a messenger RNA, and it's just kind of hanging off. It's directly being, it's directly being transcribed off of this DNA. And because inside a bacteria you don't have the nuclear envelope, you can just attach your ribosome and just immediately begin translating into a protein. So the RNA that comes off of a bacteria is ready to go. It doesn't need any post-transcriptional modification, okay? That is different in eukaryotes. Eukaryotes do need post-transcriptional modifications, and that's what we're going to talk about next. But this is an important idea to understand. So in eukaryotes, we do have polysomes. So this is what we call it. So polyribosomes or polysomes, that's what we call it when we have one messenger RNA with a bunch of ribosomes attached, okay, because they're translating. So you can see this in eukaryotic cells. However, you would not see them in this close proximity because we have to take our messenger RNA in a eukaryote, modify it, export it out of the nucleus, and then you could have polysome or polyribosome formation. Okay? In bacteria, we can do it almost immediately. Okay? So that's the important difference here. So we need to talk about something that bacteria generally don't do, which is post-transcriptional modifications. Transcriptional modifications tend to fall into three different categories. Another word for this is your pre-messenger RNA processing. Now, it doesn't necessarily have to be messenger RNA, but for the most part, that's what we're generally talking about. So we're going to say that. We're going to call it that. I like to say post-transcriptional processing. So when we're making our messenger RNA, there are three things that we tend to do. And we're actually going to talk about the middle one first. That's splicing, which is what most people have heard of. You probably have heard of a 5' cap at a 3' poly A tail as well, but splicing gets talked about a lot, and that's your introns and your exons. So let's talk about those first. So exons are what we keep, and introns are what we actually get rid of. And so we have our precursor messenger RNA sequence. Okay? It's already been capped and tailed, but we're not talking about that right now. And we want to cut out specific pieces. And so the pieces that we actually cut out are called introns. Okay? And so when we actually want to mature our messenger RNA, we cut out our introns and then we stick the exons back together, and that forms our mature messenger RNA. Now, how does this happen? Well, the process is called splicing. And the actual proteins, the complex that actually does this is called the spliceosome. And one important idea to think about is that you could do this in different ways. So I could have one giant long messenger RNA, so that's this long guy right here, and I could cut out different pieces and then put them in a different order, okay? And so let's say, like for instance, so this messenger RNA down here, I think I'm using all 5 pieces. Okay, awesome. Well, what if we lose exon number 3 for this one? Well, there's going to be similar structures, right? So we're going to have a same structure here. So this is showing what looks to be a transmembrane protein. And so what we're going to do is we're going to cut out exon 3, and that's this region right here. And so we actually have consistency in these proteins. They all share the 1 and the 2 here for this region, so this nice transmembrane region, but the rest of it can vary. And so that's called alternative splicing. When we remove different introns, that means we can take one gene that made a messenger RNA and then cut it out differently and make multiple proteins. Now, there is usually consistency there. And so if you have a series of proteins, let's say, that all have similar structure and similar functionality, but there's some differences, that's probably alternative splicing. The process of alternative splicing is really interesting. And now we call that complex of proteins the spliceosome. We love to add some to stuff when it's a big complex, but its job is to excise or cut out. Excise is just a fancy way of saying cut out your introns. Now that is controlled. It's very highly controlled. It's controlled by a variety of mechanisms. In this case, it's actually using small nuclear RNA. So that's something I didn't mention before. Briefly I mentioned it when we talked about here. So that's your Rna Polymerase 3 is going to come in and make these little messenger RNA, not messenger RNAs, they're just small little RNAs that have complex folding patterns, which we've talked about before. RNA is able to fold over on itself in lots of fun, cool ways. And by folding differently, it generates different structures, which can then bind to different things. So don't think that nucleic acids are only there to transmit information. That is not their only job. They also perform a lot of structural jobs. And so I wanted to cut to a video that actually talks about the spliceosome and how the spliceosome actually works.
Here we see an RNA molecule with a single intron. Several signals exist within the intron that are used in the splicing process. From the 5' end of the intron, these are GU, the A branch site, a pyrimidine-rich region, and the 3' AG. The GU and AG sequences define the beginning and end of the intron. Splicing is mediated by the spliceosome, which consists of several protein RNA complexes. The first step involves two complexes that bind near the GU sequence. The RNA is then looped, and 3 other protein RNA complexes bind. This final complex then undergoes a conformational change. Introns are non-coding RNA sequences that must be removed before translation. The process of removing the intron is called splicing. The intron is then cleaved at the 5' GU sequence and forms a lariat at the A branch site. The 3' end of the intron is next cleaved at the AG sequence, and the 2 exons are ligated together. As the spliced mRNA is released from the spliceosome, the intron debranches and is then degraded.
That video is a virtual cell video. They make really, really good videos. They're very, very accurate, so you could even see the ATP coming in to actually help with the conformational changes in that protein complex for the spliceosome. Really, really cool. That's from North Dakota State University. They have a bunch of videos. They're really, really good. You should check them out. Most of them are on YouTube, but they have a website as well. I think they had an NSF grant to do that. Now, why is this important? Well, I'm an immunologist, and so one of the things that you learn in immunology is how do you actually pick what genes are used to make the protein tips, if you will, of the antibodies. So here's your antibodies down here. There's an IgM, a little bit longer of a heavy chain, and then your IgD here. So these are the first proteins that get generated. And so what you're going to see is the Vdj recombination and the constant region as well there are a bunch of different pieces, so different chunks of DNA that we can grab, turn into messenger RNA. So this is our DNA up here. We're going to grab a chunk and make a messenger RNA off of that. And how we actually pick that, this is called Vdj recombination. That's from splicing and alternative splicing. So I'm picking which region I actually want to grab. Now, some of this rearrangement, it's a little more complicated than that because you can have alternative splicing of the messenger RNA. This one actually involves some splicing of the DNA, which is a different process controlled by RAG. Don't worry about that, but the concept is similar, and I just like it because it's talking about antibodies, and I like antibodies. So we're talking RNA processing, right? So that's your post-transcriptional modifications, your pre-messenger RNA processing. We've talked about introns and exons. It's just another picture I like to throw in there if it helps you figure out what's going on. But there's 3 more things that we need to talk about. So we need to talk about capping, we need to talk about tailing, and we need to talk about how to actually get RNA out of the nucleus and into the cytoplasm. So let's talk about that. So the 5' cap so it's on the 5' cap. So we look at the structure of our messenger RNA up here. So we've got our coding sequence, and then we have untranslated regions. So we have a 5' end untranslated region, and you're going to have a 3' end that's an untranslated region. Okay? So messenger RNA isn't just one little transcript that makes a protein. There's a lot going on, and we're going to cap it and we're going to tail it. Now, the cap is usually a methylated guanosine. And so it's got an extra methyl group on here tacked on. And so if your messenger RNA is here, so this is your sugar phosphate backbone, right? Because messenger RNA is still nucleic acid. And then we've got lots of phosphates here, and then we have our methyl guanosine. And so what's its job? If you take cell biology, we spend a lot of time talking about this. The main job is protection. The cell has a bunch of exonucleases, so those are enzymes that are designed to destroy RNA. So either because we want to get rid of the RNA because we don't want it hanging around, maybe there's something wrong with it, maybe we don't need it anymore, so let's chop it up. Or if a virus, or maybe a bacteria, but usually a virus, is actually inside the cell, then a lot of viruses don't cap or don't poly-A tail. And so those look very strange. Those are messenger RNAs that look weird because viruses can have RNA as their genome. And so if you encounter an RNA that doesn't have a cap, it could be a virus DNA or a viral RNA, and so you should chop it up. Okay, so that's protection. It also helps with nuclear export, which we're going to talk about in a second. It helps as a region that just gives some recognition to the actual spliceosome, so it can help it with intron removal near this end. And it also just sort of, it's all part of the complex of what, it helps a ribosome see what's going on. It needs to be oriented properly, and actually having the cap on there can help bind to a region of the ribosome that tells it, okay, hey, here is the start of the actual messenger RNA. So that's the cap. So those are some of the functions of the cap. The tail, so we call it the poly-A tail. It's a polyadenylation. So here's the actual tail. So here's our cap. This is how it actually looks when it's bound to one of the complexes. And so this is the complex that's going to actually add the poly-A tail. And so you can see we got our cap here. We've got our 5' untranslated region, so that's right here. And we're going to go down here. ORF stands for open reading frame. So that's just what's the ribosome going to actually look at. Those are called ORFs, or open reading frames. And so this is the coding sequence of your messenger RNA. So that's what's in green up here. Then there's our 3' untranslated region, and then there's our poly-A tail. Now, what's its main job? Stability, okay, as well as export. And so that's going to be the big one. And then, of course, these RNAs are going to go and get translated, and so they do bind to the ribosome at specific regions as well. But honestly, usually export is the one that we talk about, as well as stability, that's not that different from here. Export and stability. Protecting from degradation is also stability. So that's the main job of that. One of these main things I keep talking about is nuclear export. And so if you take cell biology, which I teach, we'll talk in more detail about this. The actual export of messenger RNA, among other things, from the nucleus and import of things into the nucleus is incredibly complex and involves a ton of different proteins. This is just the RNA complex, the protein complex to get messenger RNAs in and out. Okay, so this is inside the nucleus, and then it's heading out of the nucleus. It's kind of like backwards from between the two pictures, but you can see they're kind of flipped, so that makes sense. You can see there's tons of proteins associated here. You have to get your proteins associated with the messenger RNA. Some of those are responsible for actually bringing it down here. There's the PABP, that's this guy. And though some of them will traffic it towards the actual export complex, some of them bind to specific regions of the export complex. You can see all these different proteins have functions in the export complex, and it's got these little fun little wiggly guys inside. Anyway, so it's a really complex, awesome, really, really interesting world of how to actually get a messenger RNA out of a nucleus and into the cytoplasm so you can actually translate it. And so that's all the steps involved. that bacteria don't have to do. They can just skip that whole thing and just immediately start translating, which is really cool. Now, one last thing. I talk about this in biochemistry a lot, and so if you also take biochemistry with me, this is going to be a repeat of this week. We talked about nucleic acids in biochemistry, which it covers this as well, but I want to talk about it in this class because not everyone is in my biochemistry course. And so RNA interference is really, really important. So not all of our not all of the RNAs we make, actually get turned into messenger RNAs. Some of them will go on to be small nuclear RNAs. Some of them will go on to be ribosomal RNAs. But if we want to use RNA as a tool, one of the tools that we can use is interference. And so we can make we can make RNAs. I keep saying messenger RNA, I'm sorry. We can make small little pieces of RNA that can then be used by other machinery, namely the RISC. So RISC is the main one. It's going to use a little piece of guide RNA to then go and bind to other RNAs, usually messenger RNAs, and it will cause cleavage of those messenger RNAs, which then disables them. So this is one more level of control. That's what we've been talking about, is levels of control. So how we get the RNA polymerase to bind, how well it binds, how we get just different proteins associated with it. Are we going to cap them? Are we going to tail them? Are we going to actually export them? What are we going to do? How are we going to splice it? And then now we can actually use other pieces of RNA to cut messenger RNAs that we don't want anymore. So why did we even make it in the first place? Well, things get complicated. Maybe we made a bunch of messenger RNA and now our feedback mechanisms have said, hey, you know what? We need to actually cleave this. Or we could actually use this as a weapon against viral RNA because it's not that different. As long as this little guide messenger RNA, or guide RNA in blue here, matches or is complementary to another RNA, then it will cause cleavage. Dicer is what cuts them into, so it's double-stranded here, RNA, and it cuts it into this single-stranded little guide RNA that we can then use to look for complementarity. Now, there are two general kinds of RNA interference RNAs. So there's microRNA, which only needs 2 to 7 nucleotides in the guide RNA to actually bind and then cleave the protein. Oh my gosh, the protein, really? Then cleave the RNA that it's going to cleave. Or you can have really, really specific small interfering RNA. That's usually about 21 nucleotides of complementarity that's required, which is really, really high. So that means it's going to very specifically knock out messenger RNA. As opposed to microRNA can be a little more generic and knock out a bunch of a larger variety of RNAs. And so we can use RNA interference to actually stop the production of proteins. And so we can use that, your cells use them, and then we can use them in the laboratory as well. It's a really, really common technique. And I'm going to end with a video from Nature that talks about how RNA interference actually works.
Scientists have been making rapid progress in understanding RNA interference, or RNAi. Many organisms use RNAi to control genes, and it can also be used as a tool in the laboratory, and in the future perhaps as a therapy. This animation will introduce you to the principles of RNAi involving 2 important types of RNA molecule, small interfering RNAs and microRNAs. Eukaryotic cells have many sophisticated ways of controlling gene expression. In the complex environment of a cell, these mechanisms need to be precisely targeted. There's a group of mechanisms that use small RNA molecules to direct gene silencing. This is called RNAi. Inside the nucleus, most genes that encode proteins are transcribed by Rna Polymerase II. The primary RNA transcript is processed by splicing and forms a mature messenger RNA, sometimes called mRNA. The messenger RNA is then exported from the nucleus into the cytoplasm. Here, ribosomes catalyze translation of the messenger RNA to form polypeptide chains that fold into proteins. But this is also where some small RNA molecules can have their silencing effects. There are several types of regulatory small RNA. Small interfering RNAs, known as siRNAs, are derived from longer double-stranded RNAs that are either produced in the cell itself or are delivered into cells experimentally. The introduction of siRNAs or double-stranded RNA is widely used to manipulate gene expression. MicroRNAs are another type of small RNA. Most microRNAs come from RNAs that are transcribed in the nucleus, which then fold and are processed before being exported into the cytoplasm as double-stranded precursor microRNAs. The double-stranded precursors of microRNAs and siRNAs bind to dicer, which is an endonuclease protein that cuts the RNA into short segments. Most siRNAs and microRNAs are approximately 21 nucleotides long. The short double-stranded RNA then binds an Argonaute protein. One strand of the RNA is selected and remains bound to Argonaute. This is called the guide strand. The combination of the RNA and Argonaute, along with other proteins, is called the RNA-induced silencing complex, or RISC. siRNAs direct RISC to bind to specific messenger RNAs. The targeting is precise because it's determined by base pairing between the siRNA and the target messenger RNA. siRNAs often have perfect complementarity to their target sites. Once bound, Argonaute catalyzes cleavage of the messenger RNA, which will then be degraded. MicroRNAs also guide RISC to messenger RNAs. Usually only part of a microRNA, known as the seed, pairs with a target messenger RNA. This imprecise matching allows microRNAs to target hundreds of endogenous messenger RNAs. Targeting by a microRNA can lead to messenger RNAs being degraded or translation being inhibited. Argonautes and their small regulatory RNA cofactors are found in plants, animals, fungi, and some bacteria, and their importance in a multitude of biological processes and as tools continues to be revealed.